Together with Professor, as Arthur mentioned, we are representing Institute of Plant Protection, which is a national research institute, and we are dealing with the Department of Mycology. That means we are dealing with the diseases in general. So there are two, as I said, we are dealing with the diseases, so there are two good news and one bad news. The first good news, I'm gonna speak only in English. I don't want to speak in 10 languages, so it's a good news. As I said, it's a half an hour. The second good news, you're gonna see a lot of pictures with symptoms of the diseases, but the bad news is you won't be able to see these diseases outside because of the breeding guy of Tomasz and of all of the organizers. You won't be able to see these diseases. That's good, and I'm sure before I'm going to start talking about the diseases, just a short introduction. So on the left side of this chart, you see the numbers. This numbers is a potential which is closed in each and every seed you're getting for the breeding company. So in case of different cultivars, you see that from the oilseed rape cultivation we can even get about 10 tons per hectare. It's a surprise, I must say. But on the right side of the chart, you see something that we call the farmer's reality. So this is also numbers, but as you can see, it's almost a half of the potential. So you can kindly ask me, so what is the reason why we cannot exploit the full potential of the seeds? I'm sure that you're gonna find the answers during these presentations, but even on this slide, there is a tip hidden on the right corner that thanks to the diseases, thanks to the pests, thanks to the weather conditions, and thanks to the many agrotechnical factors, you are not able to get this full potential. So of course, as I said, for a different group and different cultivars, it looks always the same. So we are not able to get the full potential, even if we try and it's not so simple. Even if we are making the research, it's not so similar to, not so easy to get the numbers. So you have to always be aware that if you want to get the infection of the plants, it's always not so simple. Because there are many factors that influencing the development of pathogenic organisms. So starting with the source of the infection, which has to be on a field, for example, to get the plants infected, finishing with the biology of pathogen, of this proper stage of growth, and the weather condition once again. So you have to put all of these puzzles together to get the infection. And later on, even if you succeed, sometimes, like this year, for example, we're still waiting for the Sclerotinia on a field, and probably thanks to the weather conditions, when it's cold and it's not so, the soil is not so much more wet, that's probably we are still waiting for the Sclerotinia. But it's good for the farmers, it's bad for the researchers, if I can say so. So as you can see, there are many diseases, and some of these diseases will be visible for the whole fall vegetation period, so from the beginning, from the seedling stages, like the seedling blight. And some of them will be visible, like Phoma, like clubroot, alternaria, and many others that you can see on a chart. They can be visible for the whole vegetation period. And in the brackets, we put the numbers once again. So the numbers means the potential losses that each of these diseases, that means each of these diseases can steal your money in general. But even if we put, for example, 50% for Phoma stem canker, it doesn't mean that this disease can even get 100% of your yield. So that's the tricky thing. Even if you got a 15% for a seedling blight and many other diseases, in some cases it will happen that you won't be able to make production success because you will finish your production in the very beginning stages of growth. So every time you have to keep in mind that in some cases each of these diseases can provide you even a full cancellation of your yield. And there's another thing, because you have to always monitor your production fields, because you have to know that from the infections to the appearance of first symptoms, there is always a time. In case of many diseases, in case of Phoma, it looks you need about 2 weeks to see the symptoms of the infection. In case of Sclerotinia, for example, in the flowering stage, you also need about 2 weeks. But in case of clubroot disease, so the diseases with tumors on the roots, you're gonna need about 4, 6, and even 8 weeks. That is why it's not so easy if the farmers, they're asking us, could you please make a test for us 1 week before sowing if we got a clubroot disease? We are not able to answer because we need about 2 months to get the results of the biological test and to give these answers to the farmers. So maybe you will be able to use this information in the next season, but that's why we are not able to provide you with any data before the time, because 6 weeks is the minimum time to get the first tumors on the roots, even if we know that the soil is infected, even if we're making an artificial inoculation of clubber disease, which is possible, of course, like in case of many other diseases, like in case of Sclerotinia and many others. So you need a lot of time. And the last group, but not least, for example, is a light leaf spot and verticillium disease. You're gonna get the infection in autumn time, but you're gonna see the results, the symptoms in the springtime. So very often it's a situation that if we are making sprays in the autumn time, it's usually these sprays for FOMA or for trying to catch the growth stage of the plants. You're gonna control also these diseases. But the point is, there is a need to take a lot of time to see the symptoms. That's the point. So once again, if it comes to diseases, each and every fungi, because they are mostly the causal agents of these diseases are mostly fungi, they're gonna need some special favorable conditions. So in case of probably 90% of these diseases, you're gonna need an average temperature about 10 degrees, and later on you're gonna need a high level of soil and humidity, and air humidity. But in case, for example, of powdery mildew, as you can see on the chart, and in case of verticillium, you don't need a water, you need just the temperature. So you have to, once again, be aware that in any kind of conditions that are on a field, under the field conditions, there is always a fungi that is waiting to catch your yield, to catch your money. So that's why you have to control the diseases. That's why you spray in the fields against diseases in the autumn, in the springtime, in the flowering stage. It will be different in the different seasons because it's connected with changes in pathogens biology, like in case of Sclerotinia, you're gonna see it on the charts later on. But it's still important to remember that there's always conditions favorable for any kind of fungi. So that's why it's so important to make sprays and so on, and using different tools. So coming to the tools, we decided to divide it on 4 groups, 4 methods. So you got the names of the diseases on the left side of the chart. And thanks to the breeders, for example, we know that we can find some cultivars which has a genes of resistance to clubber disease. That's perfect, and it's working perfect. The FOMA stem canker, once again, a lot of genes, and I'm sure you'll be able to see these cultivars outside the stand. And of course, there is a tulip yellow viruses, a disease which was a few years ago named like a marketing disease maybe, but now, trust me, it's a serious disease. And even if our institute is making some tests, in this year we got information that 100% of the samples which were analyzed in our institute were positive for the virus disease. So it's a huge story behind this. So you can use the breeding method. You've got pluses in each and every spot. In case of agrotechnical factors, I'm sure that it should be another lecture, maybe just about 90 minutes about the agrotechnical factors, but we ain't got so many times, so just we're gonna put it and stay like in here. There is some biological stuff. We are dealing nowadays with many hundreds, even also biological compounds, and trust me, once again, there are many biological compounds that are really working well, but if you compare it to the chemical solutions, it is always, you have to always apply it in a proper stage. You have to keep it, for example, in a refrigerator, so you have to buy some stuff to just get it in your warehouse. So thanks to the biological compounds, nowadays we are able to manage FOMA, we are able to manage Sclerotinia stem rot, but you have to use it just before the sowing, or meanwhile the sowing, mixing the soil with the biological compounds. So you're not able to see the disease and just make a decision, let's use a biological compound. It doesn't work that way in case of biologicals. And for example, to the seeding blight, we also got some tools that can be useful. And probably one of the most important and probably one of the well-known methods, so the chemical method, you see a lot of pluses, thanks be to God. We still got enough active ingredients. Even if we withdraw, we cut some of them, even the the ones which were perfectly performed for the oilseed rape, like chloropyrifos, like thiophanate metal and so on, prochloras and many others. We still got enough to control the diseases. We also put a minus in case of turnip yellow virus, because we are dealing with the vectors. So we're using chemicals, we use insecticides to cut the vectors, not the disease. So it should be or maybe we should put plus and minus in this part. But you can see that we are able to control Alternaria, we are able to control Syndosporium, powdery mildew, Phoma, gray mold, verticillium, and Sclerotinia. As I said, it's enough, still enough ingredients. And in case of white leaf spot, you will see the symptoms in later on charts. We usually controlling diseases in the autumn time, and thanks to the sprays, we are also controlling this Pseudocercosporella capsella disease. Even if there is no registered products on the market, you also cover with a spray this disease. So that's good. Even if you don't know you got this disease, you can control it very easily, spraying, for example, triazoles in autumn time. So it's always about the yield reduction. It doesn't really matter. You can read this chart because it was prepared especially for you, but the most important thing that comes from this chart is always about the yield. So it doesn't really matter what kind of disease you got, just like from the point of view of the farmer. The most important thing is always stealing your money and always stealing your yield. If it attack hypocotyl stems, roots, pods, and seeds, it always finishing at the end with stealing of your money. So first, a disease which is well known, of course, and as I said, you will not find the symptoms outside in general in here, but it's a disease called Phoma, stem canker, and it's the spots in the autumn time you're gonna see a spots with black dots on it. So these are pycnidias, and you're probably well known about it. So later on, it is transferred to the stems. And at the very late stage of the season, you see the same spots on the stems. It is also covered with the pycnidias. And if you look on the pods, it can be also transferred to the pods and later on to the seeds. So that is why it's so important to control this disease, to spray the plants with the fungicides to avoid getting the pods infected. And if you look on a single stem, it's fully damaged. When the infection is strong, you're gonna see it also on the next slide. So one after the next one. That's the image which was taken in the autumn time. So on the first look, you see just the plants. But if you got the magnifying glass, you're gonna see some fresh lasers with the pycnidias on the leaves. So as I said, maybe it's a nice view for the phytopathologist, but it's not a nice view for someone who is cultivating for about 500 hectares of oilseed rape. And just to keep in mind this picture, you will see when the plants are without the disease symptoms. So this is the upper side of this photography. It looks quite normal, the white stems inside, but at the very low part of these pictures, you see the plants, the stems fully damaged with Phoma stem canker, so there is no chance to transfer any nutritions to produce good quality yield in case of the infections. And the same story with the leaves. You can see even 75 to 100% of the leaf area can be infected. So it's a long journey from the beginning, from the seedling to the end of the pot. So the breeding is common thanks to the breeders. We got about 75 varieties registered on the market containing some genes, as I mentioned previously. A lot of companies is delivering, about 7, as I remember, companies delivering these cultivars. And currently, that's a news just from 1 week before, there is 52 varieties containing genes which are being tested this year in the registration test. So for sure we're gonna get some new cultivars. That's good information. I'm sure Rapool also will provide you next season with some new material, with new genes or with new cultivars in this case. So it's good that we got this tool available. And as I said, the chemicals are still on charge. So hats off for the breeders, if I can say so. Anyway, the second disease, you can call it even a queen of the diseases, probably. So it's Sclerotinia, the disease that, as I said, we are waiting for as the researchers. It's still not on a field, but it will be, trust me, it will be, because every single season we see the symptoms of the disease. So it all starts with the small lesions on the stems. Later on, in the middle part of the chart, you see structures of the fungi, and at the very right side of this slide, you see the sclerosias. So it could be about 100 even or more in each and every single stem. And if you know that from one sclerosium, you can get about 20 millions of spores of this disease. If you count it 100 times 20 times the number of plants on the plantation, it sounds like really crazy. And that's why you have to spray in the beginning of the first petal flowers are falling down. But you will see that the biology of this pathogen is changing a lot. So there will be some years that even if you spray in a proper time according to the registration label, it won't be enough. So that's why it's good, for example, to mix the biological compounds with the chemical compounds. But you're gonna see it later on the chart. So this right side of the chart, you can see there are also pods can be infected, and also sclerosias can be visible on the pods. And why is so important this disease? This disease is so important because it's a polyphag. So you can find it on a sunflower, on the potatoes, tomatoes, many vegetables, many plants. So it always looks the same. It's always producing sclerosias, and it's always producing hundreds, millions of spores. So it's really serious threat. So thanks to the information we gathered in a previous years, we decided to make a few scenarios. So the scenarios of protection, what should you do in case of different weather conditions will appear on a field. So the first scenario is connected with the high amount of water, so maybe not so common situation in the time of flowering, plus high temperatures. So this is the first scenarios. You see the cracks on the stems, so something that is usually happening in the springtime when the plants are starting to grow so quick. They just like it's full of energy, so the stems are cracking. Later on, probably a gray mold will attack these stems. But so if in case of high amount of water and high temperatures appear on a field, you will see the cracks of the stems. You will see that the soil is quite moisture, and you're going to see a rapid development of causal agents of the diseases. As I said, when there is a water and there is a temperature, there is always a kind of fungi that will try to attack your plants. So we think the solution for this situation is to spray with the fungicides on the yellow bud growth stage, and you got a chance to use the preventive mode of action fungicides, but probably you're gonna have to spray a second time when the plants are in a full flowering stage, or even 1 or 2 weeks after this stage, to get the plants health. And as you see on the ground, there's a pluses and minuses, of course. So pros and cons. So pluses are you got the plants protected from the beginning of flowering stage until the pot formulation, but you're gonna need to spray probably 2 times to get these plants healthy. And so you need to spend much more money to consume your time, your energy to make this a solution. And the second scenario is probably much more common. So this is a situation when there is a long-term drought on the field, and the temperatures are mid to high. So maybe we can connect it to the situation that is nowadays on the field. So the flowering period is quite short. The soil is dry. The development of causal agents of diseases is almost inhibited, even for sclerotinia and for gray mold. So the plants are without symptoms. You won't be able to see the symptoms. So you're waiting for the symptoms, and you're still thinking, so what is the best time to use the fungicides, and how many days can I delay this spray to be perfectly applied? So the solution probably, from our point of view, is to use a late fungicide application, to wait, for example, until the rain will come, and when it will be appropriate moisture content of the soil of the earth, and maybe you can try to reduce the dose, the amount of the fungicide that you're applying. That's good. So once again, the minus is you have to stay focused, be ready to make a spray. So because you can possibly miss the symptoms, but the plus is you can make an application under the low pressure of diseases. That's perfect, and you've got some more time to make your decisions. So it's really, once again, a nice idea. So we think this scenario will happen this year. So these ones who will spray just in the next days will get the best results with controlling Sclerotinia. So this is the picture. It was taken a few years ago. It was even a pandemic time because the professor had a mask on his face, and that was the situation when we were waiting for the first petal flowers to fall down, but the plants were full of Sclerotinia. So this is, as I said, the situation where the pathogen is changing its biology. So we are waiting to make an application of fungicides, but the disease is on a field. It was about 10 to 15% plants which were looking like this, full of sclerotia. So it was probably too late to any kind of spray, and the application should happen 2 weeks before. Even if you got the information on the label that you spray in selected or proper stage of flowering of the plant. So sometimes it looks, in some years you're gonna need to spray faster, in some years you're gonna need to spray later in the season. This is a kind of a confirmation from one of our friends from Germany, a researcher who is dealing with the Clabber disease and many other diseases. She found out that in the year 2022, the creation of apothecias, so the small fungi, as you can see on the right side of the chart, they producing the spores. So the time that there was a production of the spores was quite similar to the flowering stage. So all of the sprays which were taken in 2022 was appropriate if they spray in the flowering stage. But if you look on 2021, so year before, you will see that if you spray in the flowering stage, it was about 3 weeks too fast. So it all happened, the spread of the disease, the spread of spores happened almost 2 or 3 weeks after flowering stage. So probably all of the sprays which were taken during the flowering stage were not sufficient, were not positive. So you spent the money, you didn't get the reaction of the plants, you didn't get the success. So probably farmers had to spray once again just 2-3 weeks after the flowering stage. But now it's 2025, so we know this information after after 1 year, 2 years after it happened. So that's why it's so difficult to catch the proper period of time that you can use the fungicides in a season. Next disease, it's Alternaria disease. It's also well known. It could happen that even in a seedling stage you can get the plants infected. Usually it looks like a black dots on a black spots, black dots, whatever you call it, on a small leaves, on the stems, and at the very end of the season on the pods also. Why it's so dangerous? Because you can get some of the yields will fall down from these infected pods. That's the point, and that's why you have to be aware that this disease is not so funny. The next one is gray mold. It's a disease connected with any damages, any bad things that happened in a season. So if you, for example, damage your plants with the nutritions, with the machinery, whatever you did wrong, you're gonna get an answer with the infection of gray mold. So even if, with these cracks which I showed you on the previous charts, there is always a space on the plants to get attacked by the botrytis. So you will find the symptoms on the leaves, on the stems, and once again, even if we put it that you can get a 20% lower yield, it can happen that if it will attack these cracked stems, you can get 100% of plants which are laying on the ground, and that's the serious problem anyway. So, and on the right side, you can see that also pods can be infected. So once again, really serious disease. But if you use chemicals, you can control it very nice. So it's not a big deal. A disease, next disease is really approaching to different regions of Europe. So we can call it an old new disease. We had also yesterday a conversation about this disease also. So this is a Perennial, so light leaf spot. The disease that looks like someone with the glass of milk just enter your field and just put a droplets of milk on each and every single spot, each and every single leaf on your plantation. Of course, it's not happening because no one is coming to the field with the milk in a glass in a hand. But it looks like a droplet of milk, just it's laying on the leaves. Also, it can attack the stems, and also it's a serious threat. Especially, it's connected in general with the climate changes. So for sure you're gonna find it in Germany. For sure you're gonna find it in Poland. I'm working in the Institute for about 20 years nowadays, and I must say that this disease is visible in the last 5 years. So I'm sure in the next seasons also it will be visible in much more high concentration. So you have to know that milk is not your friend in this case if you see it on a field. So downy mildew, one more disease which is strictly correlated with the first growing stages of the oilseed rape seedlings. There are no registered chemicals. That's the point. We are trying to, sometimes when it's a huge attack of this disease, we are trying to use illegally the fungicides which are registered for Phytophthora infestans in potatoes. So in some cases it works fine if you use the sprays that is usually used in a potato plantations against Phytophthora infestans. You can control this disease, but there is no registered chemicals for controlling of this Hyaloperonospora parasitica, and then once again it can damage the whole plantation in 1 or 2 weeks. So even if it's a 10 or 15% of taking your yields, it can take about 100 if it damage a small seedlings. Now one more disease, it's a powdery mildew, well-known disease. Maybe it looks fine, just a black material, white colored structures of the fungi on the leaves. It's for sure well known for those of you who are familiar with oilseed rape cultivation. You can find it on the leaves, on the stems, once again, and on the pods at the very end stage of the season. And there's one more disease, verticillium wilt. So this disease, if you take a tour outside the stand, maybe you will be lucky to find a leaf that looks like on the left side of the chart. So half of the leaf is dying, half of the leaf is yellow, and the second half is green. Yeah, because the structure of the fungi is blocking the transfer of any nutrients inside the leaf. So when it's blocked, the half of the leaf is dying and the half is still living. So it looks like this. Later on, in this stage of vegetation period, not in here, but in case of the infections, you will be able to see the wilts of the flowering plants, but it's always connected with kind of a black stripe. In here, on a stems, you will see a black stripes, and then you can be pretty sure that you're attacked by verticillium disease, which is also becoming very, very dangerous in last years and probably will be. And at the very end stage of the seasons, you are able, I don't know if it's possible to look on the picture on the right side, you will see a microsclerosis, so microstructures of the disease, and then you're for sure, you got a confirmation that you got a verticillium, and probably one of the last diseases, so the turnip yellow virus, connected with, as I mentioned, with the aphids. It looks quite familiar in some cases with the clubroot disease infections. So the plants, they changing their colors for the red, for the different colors. So it's not so simple to tell you on the first view of the field if this is a virus or this is a clubroot disease. But if you find some colonies of aphids, you can be also pretty sure that this is the virus. The same situation from the breeding, we got 95 registered cultivars. You can find the names on the chart, and about 78 are being tested this year. So it's a good information once again that in the next seasons we're gonna get some more cultivars which are resistant to these viruses. You can take pictures as many as you want. I hope you get also these papers before the slide, so go ahead. One more disease which is not so common maybe, and which is kind of a tricky disease because this looks quite familiar to FOMA, but you will not find the black dots. Yeah, it looks pretty the same. So you will find in the autumn time, you will find the symptoms. In the autumn time, looks like a phoma, but you will not find the pycnidias. And if you will put it under microscope, there is a different organism that is making this story happen. So this is Pseudocercosporella capsule. That's the name of the fungi. And as I said, thanks to the sprays, thanks to the autumn sprays with the triazoles, for example, on the phoma, you are able to control this disease also. So nowadays it's not a serious threat, but you have to be aware that it can happen, that your field will be infected with these fungi. And once again, the clubroot disease, which really looks nice for someone who is taking pictures, but but when the whole roots are just changed to the tumors like this, there's no chance to get a high yield in any of the cultivars. That's why probably the breeding is giving us a chance with the genes. This is how it looks in the flowering stage. You will also be able to find many tumors on the plants, and you will not find it outside. So as I mentioned, it's good, and thanks to the breeders, it's not so simple to get the plants infected. So coming to the breeding, we got 21 varieties registered in National Register. You can find the names on the slides, and another 16 are approaching. We've got some new genes, even Rapool, I'm sure, Arturu, we're going to talk about it on the field. There are new genes, new varieties, so it's good. And probably in the next years, we're going to get some more and more. That's perfect situation. So as an institute, we are making a biological test. It's for the testing central variety organization, and based to this biological test, we have to take a lot of samples. We have to prepare the pots in the greenhouse, and we are the only organization in Poland that is making such a test for the testing variety research center. And as I said, once again, we're going to need about 6 to 8 weeks to get the results. And of course, we got a lot of hundreds of these pots with the soil infected with the clubroot. And after this test, we are making assessments. We are counting the index of disease. And later on, the varieties, they're getting approved, for example, as a resistant to clubroot disease. It looks quite simple, but trust me, you have to find a lot of inoculum of this disease across the country, make a lot of repetitions, make a lot of physical work, digging in the ground, sowing, assessing, cleaning, whatever. So after you're getting just a number that the cultivar is resistant, but before you get the number, you have to work a lot to make this in a proper way, to be able to compare it between the companies. So it's really difficult job that is done by the researchers. And we are coming to the end of this presentation. So I found an information together with a professor that some researchers made a research connected to use of isothionol together with Bacillus amyloliquefaciens in case of controlling clubroot disease. So it looks when the inoculum was low, for example, so the gray stripes on the charts, it works quite nice. So maybe the biological compounds and this isothionol ingredient will be helpful. Now it's registered only in South America. So we are waiting to get some of this product to Poland and start our tests. But it's a small green light in the tunnel in this area. And some of the Chinese paper work, they put together all of the tools that are available in case of biological compounds to control the clubroot disease. So it looks like some Bacillus strains, some Lizobacter, Streptomyces also, when they're testing it, they are able to get some results in the area of clubroot. So it's good that not only the breeding companies is dealing with this disease, but also researchers across the world are trying to find the solutions and find a proper biological compounds to control this disease. So as a summary of this presentation, we have to be aware that in every stage of the vegetation period, as I mentioned, there is a disease that can attack your plants and can get your yield. We are still got enough tools for controlling the causal agents of diseases. That is good news once again. There is still enough chemical products registered and available, of course, for controlling diseases. The number of cultivars with genetic resistance is increasing. That's once again good news. And of course, we have to keep in mind that the genetic resistance is not permanent, so we have to use it in a proper way, not mixing, for example, resistant and non-resistant seeds and sowing together, because it's kind of a nonsense. So as a finish, I can say, keep calm and sow oilseed rape, and have a nice day anyway on the field. Thank you very much for your attention.